Welcome everyone to today's webinar. My name is Dr. Sarah Comstock, and I'm an assistant professor in the Department of Food Science and Human Nutrition at Michigan State University. I will be serving as the moderator for today's program. In today's program, we aim to provide some clarity around commonly used terms, as well as methods, both common and emerging, that are central to conducting and understanding gut microbiome science. We are very pleased to offer this webinar to you as a collaboration between three groups shown here on this slide. IAFNS, or the Institute for Advancement of Food and Nutrition Sciences, is a nonprofit scientific organization that advances food safety and nutrition science for the benefit of public health by bringing together scientists from industry, government, and academia. The Academy of Nutrition and Dietetics Research Practice Group brings together registered dietitian nutritionists with particular interest in nutrition-related research. And any RDNs in the audience with this interest are encouraged to join for additional value and benefits of membership. Finally, my favorite organization, the American Society for Nutrition's Nutritional Microbiology Research Interest Section, brings together investigators who explore interactions between dietary compounds and the microbiome. Our focus is on fundamental and translational research to address pathological conditions and preventative measures, and to enhance human performance. I'm honored to be a member of the ASN group and to serve as your moderator for today's excellent program. This webinar is approved for 1 continuing education credit by the Commission on Dietetic Registration, and access to the Certificate of Participation will be provided to participants very shortly after today's webinar. In this webinar, we'll first cover 2 presentations, and then we're going to open up for a dialogue with the speakers to answer your questions. On this slide, you will see the control panel, which should be visible on your desktop too. We encourage you to submit your questions throughout the program by typing them into the questions box. Only the organizers will be able to see your questions. All right, before we begin, we want to get to know you, our audience, and understand what your baseline knowledge might be before you come into today's webinar. So before we show you the poll question, which is going to be on the next slide, we're going to give you a few instructions which are here on this slide. So you can find the poll by either going to the address shown, pollev.com/IFNS-SConnect300, or you can use your phone and text IFANS-Connect300 to the number shown, which is 22333. We're going to give you just a second to choose one of those options, and so that you're ready when we go to the next slide to take our poll. Oh, some people have already answered. All right, so the question is, which statement best describes your current understanding of terminology and methods related to gut microbiome science? The answers range from being completely new to the area all the way to actively conducting research in this field already. All right, we'll give you a few seconds to respond and get logged in. The instructions are still there at the top of the poll screen if you had problems on the last slide. All right, we've got responses coming in. They're starting to stabilize a little bit. It looks like most of you have some sense of the basics. A few of you are somewhat familiar. For most people, this is not a completely new field to them. All right, thank you for engaging with us through that poll question. Let's get right into today's program. So as I mentioned before, our objective today is to provide some clarity around terms that are very commonly used in this field, but they can be confusing. And understanding these terms is critical to understanding the research in which these terms appear. So in addition to covering terms, we're also going to be covering some of the common basic techniques as well as emerging methods and what they can tell us, as well as some limitations of those methods. With that, I am pleased to introduce our first speaker, Dr. Vincent Young. Dr. Young is an associate professor in the Department of Internal Medicine and Infectious Diseases Division and the Department of Microbiology and Immunology at the University of Michigan Medical School, just down the road from me. Dr. Young has a long-standing interest in understanding the pathogenesis of bacterial infections of the gastrointestinal tract and the role of the normal microbiota in human health and disease. He leads a human microbiome project on the role of the microbiome in inflammatory bowel disease. Dr. Young, we are pleased to turn the platform over to you.
Great, thank you for the invitation to speak today. So yes, I'm going to be telling you a little bit about what we would consider to be the language of the microbiome. We'll go over some terms and concepts, and I'm going to try to warn people a little bit about some of the dangers of jargon here. There are a lot of terms that are being used, and there are, in some cases, very different meanings in what people have to a given term. And I'm going to kind of bring up some of the variations here. And the key point that I'd like to say is that it's important when you're talking to someone about this, that you ask questions so that you make sure that you're talking about the same thing when you use a particular term. So let me just move forward here in the slides. As far as conflicts of interest, I am a consultant for a company known as Vedanta Biosciences. They are developing therapeutics in the microbiome space. I will not be talking about any of their work or any of their products. I am an editor of the journal EMsphere, which is published by the American Society for Microbiology. And in this talk, there will be no discussion of off-label use of FDA-approved therapies. So precision in communication. My poor trainees sometimes say, Vince, you harp on us when we're giving talks, when we're even just giving group meeting, and certainly when we're writing our manuscripts. Why is that? And well, this was just before the pandemic. These are my AP English teachers from high school. And I saw them when I delivered my last in-person seminar before the pandemic kind of shut those things down for a while. And going back to high school, I was always stressed that communication and precision in communication is very important. Because when you start using jargon, this is from the Harvard Business Review, there can be problems. Jargon can be useful. Shorthand terms that everyone agrees on can help streamline communications. But if the meanings of these terms can be variable, you can get into problems. So let's start talking about the microbiome. Everyone, as judged by the poll, has heard of it and is interested in it. If you just kind of type the word microbiome into a Google News search, you'll see all sorts of things. I actually meant to look up this one because I really wanted to know how a sprained ankle can affect my gut microbiome. But everything, how do antibiotics affect the microbiome is one part that's very important for me. Looking at how the gut bacteria, this so-called gut-brain axis, and how gut bacteria might be able to affect cognitive function, that's all very interesting to me. But here, let me just close this. There you go. But from the scientific standpoint, what do we know about the microbiome? Well, if you type the word microbiome into a PubMed search, you get a lot of articles. We've talked about this. Just last year, there was almost 25, well, actually, by the time they all come in, there'll probably be 25,000 articles published that have the word microbiome in it. Little bit of self-promotion. If you want to read a little bit more about how I think about the microbiome, I published this 5 years ago. And at that time, I did the same sort of search, and I was wondering if there was a little bit of an inflection here. There were only 8,000 citations at that time in 2016, but that's back here. And as you can see, really hasn't stopped the growth of interest in the microbiome in the medical literature as well. So at the same time, this is Bill Hanich, who those of you who have been listening to NPR over the pandemic have probably heard him. He's an epidemiologist in the Harvard system who's talked a lot about COVID, but he was also quite interested in the microbiome. And he kind of published this commentary a number of years ago, saying that, microbiome science needs a healthy dose of skepticism. And part of it is because there's been a little bit much that's been proposed, over what it can do. But it's everywhere. You go to the science literature, you go to you go to the popular literature, and everyone's talking about the microbiome. And they say things like, oh, the microbiome is a forgotten organ. It actually performs very essential functions for the body. But let's back up. I've used the word microbiome so many times. I guess I could have asked for a poll. How many people really understand what is meant when someone uses the word microbiome? So let's start right at the beginning, okay? I'm going to tell you the definitions that I am going to use. And they're not the definitions that everyone uses, but I always put this at the beginning of all my talks. I usually don't put the picture of the Sound of Music, but I have these words. So microbiome, for me, the term microbiome is referring to the total community of microbes. And although we usually focus on bacteria, this also includes fungi and viruses that affect bacteria and archaea, as well as viruses that can affect the host. But it's not just the community of microbes. It also includes the environment that they inhabit, such as the soil, the human body, or the interest today, the gut itself. If I'm simply referring to the microbes themselves, I tend to use the word microbiota, small life, a group of microbes that are living in a given environment or location. So this is not necessarily the most common use of the word microbiome. A lot of people use it kind of microbiome and microbiota interchangeably. And like the Human Microbiome Project basically referred to the microbiome as the collection of all microorganisms living in association with the human body. That's how they define the human microbiome. So this kind of end word ome, some people say from genomics and proteomics, and we'll go into those terms. Well, so some people say, well, what the microbiome is, it's the microbes themselves and all of their genomic elements that are in a given environment. Okay. Some people also use another term sort of related to ome, which is sort of the collective genomes of the microorganisms. So they're not actually even talking to the microbes themselves. They're really just concerned with the sum total of genomes in a particular environment. Who cares about what microbes they're actually in? So that's kind of confusing, right? Depending on who you're listening to, they might be using the word microbiome to refer to microorganisms, the microorganisms and their genes, or purely just the genes in the in a microbiota in a given place. So there are many, many, many papers that have been put out. This one came out a couple of years ago, and they did like the idea that the microbiome was the microbiota, as I defined it, all the microbes, plus their theater of activity. So in this case, the word is not ome as in omics, it's biome as in, an environment. So the theater of activity can be characterized by looking at proteins through proteomics, lipids through so-called lipidomics, or polysaccharoidomics. You can use genomics in here. So I like to use the idea that this is a small biome, a small, reasonably well-defined habitat with distinct biophysical chemical properties. And that's important to me because as someone who's studying not just who is there, I want to know what they're doing. I'm not just interested in their anatomy, I'm interested in their physiology. What is their function? And in the next talk, you're going to hear about lots of different ways that we can do this. But many of these involve very advanced, highly technical processes, but cultivation is very important as well. So why are we interested and why do I like to think about it that way? Well, because there are many functions that we give to the microbiota, both catabolic as well as synthetic functions of the microbiota, and of course, the interactions that the microbiota can have with their host, generally through the epithelium and the immune system. So let's go to some of the other terms. Here's another term that people hear about, dysbiosis. I used to tell a joke, but then it got kind of old, is when someone asks, well, what exactly is dysbiosis? And I would say, well, most I can tell by some of our 16S is that dysbiosis is different than datbiosis. It's not clear what people mean, but some people have defined it, such as a compositional and functional alteration in the microbiota in individuals with disease compared to healthy subjects. That's a good definition, but we don't exactly know what is normal and abnormal with regards to a person, with regards to their microbiome. Someone could have heart disease. Does that mean they necessarily have an altered or dysbiotic gut microbiome? So the other key point there is when you're looking at these kinds of associations, you have to really avoid linking any given association with causation. So that's something that we have to think about. So the term dysbiosis usually means some sort of community structure or functions that's associated with disease, but not necessarily causative of disease, okay? So another term, one of the sort of related to this is diversity, because many times you'll read that low diversity is a sign of dysbiosis. Perhaps, and this comes from macroecology, where people want to talk about different kinds of diversity at different levels, at the levels of a whole ecosystem, at the level of individual species, or within the species, at the level of genetic diversity within a given species. In terms of some of the things that we have borrowed in the microbiology field related to diversity, we do kind of refer to so-called alpha diversity metrics, and I won't go through the whole laundry list. I think I put some of them in my review. There are many reviews about these. But this is basically a measure of diversity that's intrinsic to a community. For example, if you're looking at a forest, the diversity in terms of trees is the number of different species of trees that are in there. But you can also bring in other things with alpha diversity. It could mean not just the number of different species, well, the relative abundance of species that are in there might be in a different diversity metric. And then people also talk about beta diversity, which is a comparison between two or more communities. In other words, oh, this community compared to this community has a beta diversity metric of some number based on some sort of metric that has come out. So let's go back to this idea of looking at structure and function. And again, we have all of these types, and we'll hear about it in the next talk about what are some of these techniques that are being used to study the microbiota and microbiome. And many times these have the term a meta something or other. And remember, the prefix meta usually refers to something that is more comprehensive or transcending. So a metagenome is not just the genome of one organism, but the metagenome of a microbiota are all the genomes summed up together. Similarly, the proteome is all the proteins that are present in a microbiome. And it's important that you use the microbiome, or for me, it's important to use the term that the microbiome is not just from the microbes, they actually come from the host, because some of these proteins are going to be host proteins. Similarly, with if you're looking, most of the times it's not a metametabolome, they just refer to a metabolome, but the metabolome is the sum total of all the metabolites that are present. And when you're doing metabolomics here, and again, some people use the term metametabolomics. I don't know, I think it's a little redundant, but I understand what they're saying. This is an interesting paper if you want to download it. It's from the National Academy of Sciences. Actually kind of a booklet that's talking about the human microbiome and health risk related to environmental chemicals there. So I wanted to try, this was not meant to be a comprehensive list of all the terms. Within some of the references that I gave you, and I believe this talk will be available later, so you can kind of look back to see what they are and look them up. There are a lot of more precise definitions, a little bit more comprehensive list of them. But I really wanted you to understand how to begin to think about all these terms that are related to microbiome research, the microbiota, and how it may impact health and disease in people. And the final slide that I'd like to put up is this idea of why is this important? Well, you might be familiar with this All of Us initiative from the NIH, where they're trying to sequence the genomes of several million people of various diseases, various ages, et cetera, et cetera. because there was the promise from the Human Genome Project of this idea of host-centered, so-called precision medicine. If we understand the genomes and also metabolites and immune function of people, can we assess how the host is and how the host has susceptibility risks for diseases, how they might react to certain kinds of treatments, both in a positive manner as well as a negative manner, to come up with customized therapy. But I'd like to propose, and this is from that BMJ article that I pointed to in the beginning, if we could bring up some sort of analysis of microbiota structure and function, perhaps we can actually combine that with understanding the host to truly get customized therapy to revolutionize how we maintain health and treat disease in the future. The work that I'm talking about, a little bit, the work in my lab certainly has been funded by the NIH, in many cases through these large cooperative agreements where we are bringing up a team science approach to science. All the people in my lab, former and current, who have contributed to all this work, and my collaborators in the past. And I thank you for your attention, and thanks again to the organizers for inviting me to speak.
All right, thank you so much, Dr. Young. Beware the jargon, very important. We've covered a lot of important topics, and I can see we've stimulated a number of questions in the Q&A box. I also wanted to remind you that that is available to you at all times, so you can type your questions right in there. However, we won't be covering questions until after the next talk, which is from Dr. Roeger, and he will be focusing on methods. So Heinrich Munch-Roeger is an associate professor in the Department of Nutrition, Exercise and Sports at the University of Copenhagen, and the group leader of the Microbiome and Metabolomics Research Group. Heinrich is specialized in combining the fields of metabolomics, gut microbiome, and human nutrition with the purpose of studying diet microbiota interactions in human intervention and cohort studies. He holds a PhD from the Technical University of Denmark. Dr. Roeger, we turn the platform over to you.
So thank you very much, and good morning, good afternoon, wherever you are. It's my pleasure to be able to participate, and thanks for the invitation to talk. So I'm based at the University of Copenhagen, as briefly introduced, and I'm going to touch upon some of the fundamental methods that we use in the microbiome space, and also touch upon a few of the emerging methodologies. And first of all, I have no financial disclosure or conflict of interest, so we can go straight into the topic. So basically, I would claim that we have one of the world's most uncovered ecosystems in the gut. And it's uncovered because it's difficult to assess. It's complex because it consists of bacteria, virus, fungi, and in very high density and very high diversity. And more to that, we also know that there are environmental factors and conditions throughout the gut, going from the stomach through the small intestine to the large intestine, is changing. So for example, pH, oxygen levels, substrate availability is changing, and is also unique for each individual. So that means that each one of us has a very unique and personal microbiome, and that complicates the whole field tremendously. We also have the complexity in terms of that our microbiome in total encodes for a very large number of metabolic products. So their metabolic potential is huge, and that means that all the foods, drugs, and environmental pollutants that might enter into our gut can be metabolized by our microbes, leading into a lot of different metabolites and small molecules that potentially can affect our health. So one of the key challenges is that our sampling is mainly, we mainly rely on feces samples, stool samples. And you have to realize that a stool sample is maybe not reflecting the whole intestine. It will mainly reflect the large intestine or the distal part of the large intestine. But we think that the stool is probably the best and most accessible sample we have currently, and then is somehow reflecting an average of the whole ecosystem, although the field is really needing other sampling techniques so we could have samples throughout the whole intestine. So if you have clever ways to come up with this, that would be perfect. As briefly discussed also before, we can study the metagenome. So that gives information about the metabolic potential, which genes are there, what could potentially happen. We can also do transcriptomics, so studying their somehow their gut activity, so what is likely to happen. However, there are some problems with transcriptome. I'll come back to that later. And then what is mainly used currently to try to study gut microbial activity is to focus on the metabolome, which is their microbial and host and dietary metabolites that are found in a stool sample. And that is a bit closer to what is actually happening. This can also be measured in human blood and urine, for example. So when we study their gut microbiome today, we are often left with a fecal sample, and we can of course try to isolate bacteria, and that is what traditionally have been done for many years, so culture dependent characterization. But we also have, with new technologies and the new revolution in terms of their sequencing techniques, we are now able to also characterize the microbiome by culture independent characterization, and that depends on being able to extract the DNA in the stool sample and then use different methodologies. So if we first look at their culture-dependent characterization, that is not a new thing. So since Anthony van Leeuwenhoek he was called the first microbiologist, he invented the microscope, which enabled us to look at bacteria in the microscope, and later Robert Koch and others also managed to grow and isolate bacteria. So we've been able to look at bacteria, cultivate and isolate them for a really long time. But the problem is that although we can grow them in the lab, easy to grow does not mean that they are important or abundant in the human gut. So this guy, for example, is Faecalibacterium prausnitzii, which is an anaerobic bacteria, so it doesn't tolerate any oxygen at all. And different requirements for nutrients and the environmental factors makes it very difficult to culture some of our microbes of the human gut. Nonetheless, there are today large efforts in trying to isolate the anaerobic microbes from the human gut, because we are still depending on being able to really study the microbes and their activity. So in the end, this is still also very important for the field. But the main reason why this field has advanced a lot during the last 10 years is due to their culture-independent characterization, where basically we extract the DNA of bacteria, so the bacteria is floating, the DNA is floating around inside their bacterial cells. And if we can get the DNA out, then we can profile it. And very briefly, the principle could be like this, that you add a bit of feces to a tube, and you add some buffer, and then you put in some beads and you mix it very hotly to basically break the cell walls of the bacteria and thereby release the DNA into the tube. And then you do a few steps where you try to clean out the DNA, and then you end up with some extracted DNA. This DNA can then be further characterized, and we can ask who is there in terms of which microbes are there and so forth. So we have different technologies for this. We can do quantitative PCR. This is a very targeted method where we quantify specific bacteria of interest. We could also do 16S profiling, also called 16S rRNA gene sequencing. And the idea is basically that we amplify and we sequence specific bacterial marker genes called the 16S gene, which you find in all bacterial species and also archaea. And this marker gene can be used to basically distinguish between certain types of bacteria. The final general method is to apply shotgun metagenomics, where we basically sequence all the genes that we find in a stool sample. And that not only gives us the possibility to ask who is there, but we can also ask what are their genomic potential, because we're not only looking at marker genes, we're actually looking at all the genes that are present in the stool sample. So the difference is that the shotgun metagenomics creates a lot of complex data, big data sets, which require much more expertise compared to the 16S data, which is less complex and in some way more straightforward. However, the information is also, you get more information from the shotgun metagenomics. So I tried to put up some of the strengths and limitations here, and I think I've covered them already, but very briefly, the 16S is widely used, it's relatively cheap, and the data processing is relatively straightforward, but it only targets bacteria and archaea, and you cannot tell you the functional potential. And also, very importantly, you cannot distinguish the bacteria often further down than genus level. So in terms of taxonomy, it's difficult to distinguish between the different species and even strains. So species and strain level annotation can more easily be done using shotgun metagenomics. And you also get the functional information from that type of data. And you can also get information on the viruses and eukaryotes that are in the stool samples. But it's more expensive. Data processing is complex, and it requires expert knowledge. Finally, we have the qPCR, which is mainly used for validating other findings, for looking at specific groups. It's more sensitive, and this is something that is sometimes done to validate some findings obtained from 16S gene sequencing, but you do not get the whole microbiome profile. So what are we interested in when we study the gut microbiota? Well, we are interested in who is there. We are interested in what are the abundances of the microbes. This is typically reported as the relative abundance, but it can also be reported as absolute abundance. And in that case, the relative abundance are sort of adjusted by the total number of bacterial cells in the stool sample. So that can be measured either by qPCR or flow cytometry, where you basically count the number of cells. We can also ask what's the prevalence of the microbes, how many persons have this specific microbe, what is the diversity of microbes, so how many different, and also the distribution of the microbes, what's the functional capacity, and what is the activity of the microbes. So, for example, this is typically when we look at who is there, you see there at genus level, which is the taxonomy level here that you get from 16S sequencing. Typically, you can see that the percentage of the different genus groups here. You can also look at the microbiome at phylum level, and then we are at this level, and then you get profiles that looks like this. We can also look at the functional capacity. So here you can see different functions of the microbiome. And this plot over here basically shows that although the microbiome composition in terms of bacterial groups varies tremendously from individual to individual, if we instead look at the functions in their microbiome, then it seems like that the functional redundancy and the functional differences are not really big from individual to individual. So you see that despite that we have very different composition, the functional potential is very similar, at least in this group of people. So the field is currently moving beyond studying the composition and more focusing on studying the activity of the microbes. So for this, we can use metatranscriptomics, which measure microbial mRNA, but also host mRNA in principle. So what is likely to happen? But this is mainly used in animal and in vitro models, where you can very quickly take a sample and basically freeze it. So the problem is that the half-life of mRNA is typically something from seconds to 1 hour. So this mRNA is very rapidly degraded. So it's not really a suitable technique to use for fecal samples. We can also look at the proteins. So look at all the proteins that are in a stool sample, and we can look at their metabolites by metabolomics. Another way to get a bit closer to the activity in the intestine can be obtained by, for example, using this smart pill motility capsule. And last year I tried it on myself. So basically it's a capsule you swallow, and then you carry around a receiver, and this receiver receives information from the smart pill from the intestine when it travels through. So this is the profile, and this yellow-green curve is the pH throughout the intestine, and you can see when the capsule enters the stomach, pH drops, and then it increases when it goes into the small intestine, and then it drops again when it enters the colon, and then it goes a bit up and down until it is excreted. And this information on pH throughout the intestine can basically give you some information on the levels of colonic fermentation that goes on, because fermentation is closely linked to pH. So this is a way to get a bit closer to what is actually taking place throughout the different segments of the gut. So in this field, we have many studies that show association. So basically, this is just one example where we have different food groups, nutrients, and dietary patterns, and they are all correlated with different bacteria. And the key point here is basically that we need to move from associations to causalities. So that can be a little difficult to do in human studies. At least it requires human intervention studies, which are expensive. So often we use models to deduce causalities and mechanisms. So we can do in vitro fermentation models where we try to ferment. It can be certain substrates with fecal material in the lab. We can use in vivo models where we investigate how specific microbes behave in animal models to get a better idea of how that could affect digestion, but also the physiology of the mice, for example. We can also do computer models and silico models to model how it could be how specific microbial metabolites interact with host receptors. We can study the effect of microbes or metabolites on host cell lines. We can study how it affects the intestinal barrier function in other transwall epithelial cell lines. Now we're also getting closer to studying organoids, so stem cell derived epithelium, where we basically can test how specific microbes and metabolites influence our physiology. And finally, there is also new development in organ chips where you try to mimic some of the chemical and physicochemical gradients that we find in the gut, colonize it with the microbiome and study what goes on in this very small scale, where you also try to have epithelium and so forth. So basically also as a way to reduce the number of animal use for this line of research, and also increase high throughput. But this is still very complex and challenging to do in a high throughput manner. Then I would point towards the use of germ-free animals as being very instrumental for this field, and it has really been key to advancing the field and demonstrating causalities and mechanism. And the key thing is that these germ-free mice, typically, they are basically mice that live in sterile environments, so environments where there are no microbes. And what typically is done is that either a single strain is introduced into a mouse, then the mice are called monocolonized. We can also introduce defined consortia of microbes, so multiple strains. These mice are typically called gonotobiotic models. We also can introduce whole human donor material. So in that case, they are sometimes called humanized mice. Here it's important to notice that although we transplant the whole feces, it's not all microbes that basically from the humans that do colonize the mice. So in the end, it's not comparable 100% to what we find in the human intestine. And finally, we also have conventional mice, basically where they have a normal microbiome, but the mice really depends on the vendors. So depending on the vendors, you have very different microbes as well. So this is an important note as well. But these germ-free model has been key to demonstrate that if you take microbes from, let's say, obese and lean individuals into germ-free mice, although the mice eat the same amount of food over 14 days, when looking at their body fat percentage after these 14 days, this study in 2006 found that the mice who received microbes from the obese individuals were better in harvesting energy and therefore increased their body weight. So this way of thinking has been really instrumental for the field. So my final point is basically that in order to advance this field, we need to combine human studies, in vivo studies, and in vitro studies. So for example, studying how diet affects the microbiome in humans, which metabolites are produced, what are the metagenomes of the microbiome, and associate these things to figure out which metabolites are associated with which microbes, for example, then culturing the different microbes in order to identify the responsible genes for the production of certain metabolites, and then ultimately take that species that produce their metabolite and knock out the gene that is responsible for the production of this metabolite, and then, for example, introduce a wild type strain or knockout strain in animal models to demonstrate what happens if we knock out this specific function that is responsible for encoding a specific enzyme, for example, and then in animal models test the mode of action in order to get a bit closer to the mechanism. And as an example, we recently published a study where we showed that breast milk via the human milk oligosaccharides that you find in breast milk, they are known to promote specific bifidobacterium in early life that are really dominating breastfed infants. And we found that these particular infant bifidobacterium species, they have an enzyme or gene which codes for an aromatic lactate dehydrogenase genes, which make these bifidobacterium species able to produce their aromatic lactic acids. And these lactic acids can interact with two different receptors, which are important in intestinal immune function. And we think this might be important in early life. And in this study, we used more infant cohorts. We used microbiome profiling, both 16S sequencing and qPCR. We did metabolomics, and we did microbiology. We grew different bacteria in the lab. We knocked out genes. We did enzyme kinetics, and we tested how metabolites interact with cell line receptors in cell lines, and also performed studies on human immune cells. And finally, we also used germ-free mice. So basically, this exemplifies that we really need different expertise and different models to get to advance the field. And with that, the choice of methods in gut microbiome research really depends on the research question. So sequencing of the microbiome have been instrumental in advancing the field, so 16S sequencing and shotgun metagenomics. But now the field is moving beyond studying the composition to focus more on the activity, that, for example, by metabolomics. In the end, we'll need to combine several methods in vitro, in vivo, and human studies to advance the field and prove the causalities and understand the mechanisms. Some of the emerging technologies are smart capsules, so both for sampling but also for measuring different things throughout the gut. It's got a chip and intestinal organoids for studying host microbial interactions, and also cultureomics, so the effort of being able to isolate and cultivate anaerobic microbes originating from the human gut. And with that, thanks for listening in, and I'll be happy to take any questions if you might have some. Thank you.
All right, thanks to both of you for those really clear presentations on terminology and methods in microbiome. We're now going to shift to the Q&A period, so we're going to invite both of the speakers back to the live front platform. Great. I'm going to open, actually, because I'm also personally curious about this, with a question for Dr. Roeger. Someone asked about the smart pill, and they're wondering if you could explain, is it attached to a smartphone? Can anyone access these? Like, how do we get access to this awesome technology?
Yeah, so that's a good question. So first of all, it's not accessible to everyone. So it's used for experimental research, and it's used in the clinics. Basically, it's a capsule which sends a signal to a device that you carry around, that you need to carry on your own body all the time, and it cannot be too far away from your stomach, basically. So we use that in our research to understand a bit more about how the environment in the gut might be important for the interactions between diet and microbes. So that's our specific interest in this.
All right, so it's not connected to a smartphone, it's connected to its own receiver device.
Yes.
This one's for Dr. Young. Can you clarify the difference between microbiome and metagenome? Is the metagenome involving human and microbiota genomes, while microbiome only refers to the microbes genes?
Well, again, it all depends on what terminology you want to use. It could be any of those. And so the best way to answer that is, if you're unsure when you're talking to someone, ask them what they're talking about, right? Because someone can just throw out the word, well, I was looking at changes in the microbiome, about all the different ways that could be. It could be they characterized the microbiota by looking at metagenomes, or they could have used 16S, or they could have tried to use semi-exhaustive culture. Well, okay, those are all things that they were looking at to look at the microbiota. And depending on what your question is and what you're looking for, you'll pick any one of those. And that's probably the best thing to do. I could define it however I wanted. And I could have said, yes, it could be any of those. That's the important thing.
So that leads to a follow-up question where someone asked if there's an organization that provides sort of the definitive definitions for these terms.
No. There have been a number that have been proposed, though. But as in anyone who proposes some sort of quote unquote standard, they're just sort of guidelines, actually. You're allowed to publish every which way. Journals will have people define it another way. So I said that I'm an editor for a journal, and if I get a paper where I think their use of a term is shifting, I ask them to just define it at one point.
That makes a lot of sense.
Just for the benefit of the reader.
Yeah. Let's ask a methods-related question to Dr. Roeger. Was the list of functional capacities that you provided in the one example slide comprehensive, or are there functions that are undocumented? How much do we know about the metabolites and functions of our commensals?
So I think we know very limited. I mean, their functions are limited to what we can match the genes we sequence against the databases. So there is a lot of functions that are unknowns. When it comes to the metabolites, we are also very limited in our understanding of the metabolites. So maybe currently what's most studied is short chain fatty acids, and we have known them for many, many years. But in reality, there's probably hundreds and thousands of chemicals that microbes produce on a regular basis all the time. So the challenge here is to identify the new unknown metabolites, and that's a lot of work to do, but it's needed. And yeah,
sort of related to that. Yeah, sort of related to that. It's not just what the microbes produce; it's also what they convert. For example, another common metabolite that I tend to study are bile acids. Well, bile is produced by the host, but then it gets modified by the microbes into secondary bile acids that we actually have receptors for, even though we can't generate them ourselves. Same way that they're receptors for butyrate, which we don't generate ourselves. We're using that to have communication between us and our microbes. So it's a pretty interesting and complex crosstalk that you have between our microbes and us.
And that brings us to this question, and I'm going to start with Dr. Young answering. Can you speak to a few specific knowns about the gut microbiome and its relationship to diet and health?
What's known? Well, what's known is that microbes, in terms of diet, for example, my colleague next door to me here, Thomas Schmidt, looks at so-called resistant starch. Okay, so these are complex carbohydrates that we can't digest, but if we ingest them, and they get converted by having the right microbes in the gut, which can include that Faecalibacterium that Henrik talked about. That can produce butyrate, which has good effects in terms of anti-inflammatory, in some cases anti-proliferative responses. So how that exactly affects the health of an individual, hard to predict, but in general populations, we can kind of understand some of the beneficial effects of having short chain fatty acid production be higher, and our ability to potentially boost it by ingesting more resistant starches of various sorts.
Dr. Roeger, you have any knowns to contribute to the audience?
Yeah, I think another example is in the infants, where we have their human milk oligosaccharides, which we know is kind of working as a specific substrate for the bifidobacterium. So we know that bifidobacterium abundance is highly correlated with the child being breastfed. So the links to health, in epidemiology, we know that breastfeeding is protective for infections, for example. But to what extent that goes via the microbiome and the bifidobacterium, that's not 100% established. So it could be, of course, be many other reasons, but I'm pretty sure that the human milk oligosaccharides are going to be and is important for the child health.
All right, let's go back to Dr. Young. What's your opinion on consumer tests for microbiome? Are they a waste of money, or do they offer helpful information?
Are they a waste of money? They're only a waste of money till you bring them to your physician and say, what should I do with this? How should you change my medications, doctor? Should I do this? Should I get tested for that? I gave a talk for the American College of Physicians, the largest collection of internists in the United States. And there was a talk on the microbiome, and I asked how many people had heard of the microbiome. Everyone laughed, raised their hands. I said, who feels comfortable with the microbiome? A lot of hands went down. I said, who has gotten a patient bringing a microbiome test into them? And a lot of hands went up again. I said, who knows what to do with those tests? All hands went down, and I pointed out my hands down too. It's interesting. It's kind of like 23andMe a little bit. It's kind of fun to kind of see where you are, and there are certain trends that you see. Oh, people who eat this tend to have this kind of microbiome. But we, as pointed out, both of us pointed out this thing between association and causation. Until we can lock down causation, and until we can intentionally manipulate and know that we're manipulating for good, we don't know what to do with these yet. So if you like having fun and you like sending oral swabs, skin swabs, fecal samples, whatever, to one of these companies, you can get a microbiome profile. In many cases, it's 16S based, as we heard about. But what to do with that, I wouldn't say that you can necessarily change your lifestyle based on any solid evidence by having the results of one of these tests.
All right, and so I'm going to direct this next question to Dr. Roeger. So related to that, we know a lot of causality information from mouse models, but what's the degree of translatability from these animal experiments to humans?
Yeah, so that's an excellent point. So the problem both with using germ-free models and also using mouse model in general is that they have, for example, a very different immune system. So the translation is definitely a problem. I still think we learn a lot from mouse models, for example, on fundamental things regarding how things are fermented, which metabolites are produced, and how they could potentially interact with our physiology. But it's not the final proof that it is that we establish causality. I would say that. But I think, and that's maybe also a little sadly, that the microbiome field and some of the big journals within this field have also advocated and a long time have had a tradition for asking for animal models and sort of pushing towards that is really needed. But I think we can really question how much value they actually bring in the end. I mean, it's at least not an ultimate proof that you establish causality. Mm-hmm. Vincent, please add on if you have things
No,
to add.
I totally agree with you there. We've used animal models a lot. What we try to do in our lab, and it's very similar to what you heard there, is start with people, move down to animal models, move to more reductionist systems like bioreactors and enteroids, and move up and down this scale of, again, more jargon, but it's often referred to as systems biology. Look at all components varying at different time and spatial scales and try to integrate things. And when things are conflicting, you learn something, as well as when they're in agreement. So it helps you understand both your methods for doing science and as well as what is the basic biology that's underneath. So it's fun. I think hopefully one thing you can hear from all of us on the panel is that we really enjoy thinking about and studying this particular field. So.
Absolutely. That brings us, I think we're up to our last question. So I'm going to pose this to both of you, and we'll start with Dr. Young. What type of human research studies are needed to connect specific components of diet to microbiome to health?
Yeah, so that's a very important question, and it gets back to what is your basic biologic question. Are you talking about maintaining health, preventing a specific disease, treating a specific disease? So depending on what you have, the types of studies are ones that really are borne out by preclinical medicine, so that you know that you have a principle that makes biologic sense from the point of view of the microbes in particular, and then try to extend that into various sorts of initially observational, but then ultimately, hopefully, clinical trial type, therapeutic types of trials. And there are a number of those that my colleagues and I are conducting, for example, trying to give resistant starch. Does that decrease the incidence of certain kinds of diseases in patients who have had stem cell transplants for cancer? That's the type of studies that are going on now. So they're kind of moving from the bench to the bedside, but then going back from the bedside back to the bench to make sure that they're seeing robust biologic effects.
Dr. Roeger?
Yeah, I think we are very interested in understanding why people respond differently to the same type of food. So I think that is key, and the way we approach this is that we try to emphasize more studying longitudinal within individuals. How do the microbiome change over time, depending on the diet? So instead of looking across individuals, we focus very much to look within individuals, because we have this huge variation from individual to individual. And then I think if we could find new ways to get samples throughout the intestine, that would also revolutionize our field, if that's possible. So I think that is sort of key for moving forward.
Excellent, excellent. So thank you both. That brings us to the end of our time today. I want to remind those in the audience that you can access your CEU certificate through an email that's going to come to you after the close of the webinar. Thank you to everyone that participated, offered the great questions, and even bigger thank you to Dr. Young and Dr. Roeger for taking time to share your insights with us. I learned something today. I'm sure even the experts in the audience have learned something today. It's been wonderful. Please register for our next webinar in the series on the IAFNS website. It is scheduled for April 21st. And I hope that everyone out there has a great afternoon, evening, whatever comes next for you. Thank you.